Hello everyone and welcome back. This is lecture 2. We'll be wrapping up chapter 1, A Brief History of Microbiology, and chapter 2, How We See the World. Long ago, the predominant thought was that diseases were caused by supernatural forces. In 36 BC, Marcus Terentius Varro was the first person to suggest on record that living things that we cannot see may cause disease. In 1665, Robert Hooke developed a powerful microscope that allowed him to observe living organisms and coined the term cell. By 1675, a merchant by the name of Antony van Leeuwenhoek was able to polish lenses to a high degree and allowed him to observe microbes that he called wee little beasties for the first time in rainwater. Along the same timeline, in 1668, a man by the name of Francesco Redi was the first to provide evidence against a term called spontaneous generation. We will cover spontaneous generation in more detail in the future. But first, I was able to find the quote from Marcus Varro. In his book it reads, "Precautions must also be taken in the neighborhood of swamps, both for the reasons given and because there are bred certain minute creatures which cannot be seen by the eyes, which float in the air and enter the body through the mouth and nose, and there cause serious diseases." "What can I do," asked Fundanius, "to prevent disease if I should inherit a farm of that kind?" "Even I can answer that question," replied Agrias. "Sell it for the highest cash price, or if you can't sell it, abandon it." Now we don't know where Marcus Varro got these concepts from, but it is interesting to note that the characters in his book were so confident about the existence of microbes, their causing of diseases, and their transmission that they were willing to tell Fundanius that if he inherited a farm in a swamp, that it would be better to abandon it than to keep it. About the same time that Robert Hooke and Antony van Leeuwenhoek were examining microbes under microscopes, there were two competing hypotheses about the creation of living organisms on Earth. Those two hypotheses were biogenesis and spontaneous generation. Spontaneous generation was the older of the two. It was the hypothesis that living organisms can be generated from non-living matter. And this was a matter of observation of inanimate organic compounds and the creation of life from them. For example, if you leave meat out, eventually you might have maggots that appear on the meat. And this is how people explained how those maggots appeared. The second hypothesis, biogenesis, assumed that all living organisms are derived from other living organisms. So Francesco Redi had one of the most famous experiments to test these hypotheses. Now, Francesco Redi believed that biogenesis was the answer to how living organisms are created on Earth. And so he set up an experiment, 3 different experiments, to test this. So we had a set of 6 jars, and within those 6 jars he put a piece of meat. And over the first set of jars, they were uncovered. The jars were uncovered, so flies could fly into the jars, lay their eggs on the meat, which were microscopic and weren't visible, but their maggots would then appear. In the second set of jars, in the middle there, Francesco Redi put a piece of gauze over the top. So the flies would lay down, they would lay their eggs on the gauze because they could smell the meat beneath it, and the eggs would hatch and you would get little maggots on the surface on the gauze there. And then the third set of jars on the top there, he put a cork on the jar. And naturally, with the cork there, no flies could get in. The flies couldn't smell the meat, so they wouldn't lay their eggs on the jar lid either. And so you had no flies and you had no maggots. So in this way, Francesco Redi demonstrated that the maggots came from the flies who were laying their eggs on the meat. And therefore, lended evidence against the concept of spontaneous generation. So this was a very cleverly designed experiment. As we can see, we have a positive control. That is, the jars without the lids should have a positive result. That is, they should have maggots growing on the meat. He has a great negative control with the corks. With the corks in place, there should be no results because there's no flies allowed in. And then the second one with the gauze allows air to go in and out. And the reason why that's important is because for a while, scientists or individuals said that what allowed spontaneous generation to occur was something in the air. It was some magical essence in the air that allowed it to occur. So the gauze allowed air exchange, but it did not allow flies to be exchanged. However, this wasn't good enough, and this just goes to demonstrate how rigorous science needs to be in order to go from a hypothesis to a theory. The reason why this wasn't good enough is because this is just testing for flies, and we see all over the world now with these experiments from Antony van Leeuwenhoek and Robert Hooke to back them up, that there are also these microbes that exist in cultures. And so people can notice that if you leave food like meat out, you can also have microbes starting to take over that meat as well. So people who were in the spontaneous generation camp weren't all moved by this experiment. Okay, so this was great for Francesco Redi. To lend support to his experimentation, Lazzaro Spallanzani lended more evidence against the concept of spontaneous generation with his boiled broth experiment. So as we discussed with the meat, eventually the meat will spoil. There will be microbes that grow in it. And this was one of the qualms that spontaneous generation people had with the idea of biogenesis. So Lazzaro took a broth, put it in a flask, and boiled it for 1 hour vigorously. At the end of that hour, essentially that broth was sterilized, and if left in the open air, again, it would grow a culture inside of it and spoil. Then he took a second broth and he boiled it again and had a stopper over it and pressed that stopper in after it ceased spoiling, and then let that sit out, and no bacteria grew. There was no culture that grew inside of that broth. So his demonstration was to show that there's no life-giving force inside of the medium itself or the material that the life grows on. However, this was not good enough for the spontaneous generation people or critics of biogenesis in general, because they said, well, what if the life-giving force is actually in the air? So, a man that you're probably familiar with, at least by his name, Louis Pasteur, all the way in 1861, definitely disproved spontaneous generation with his famous swan-necked flask experiment. And if we watch this timeline, we see how long it took to finally put the nail in the coffin for spontaneous generation. And this is how the experiment was conducted. He followed Lazzaro's experimentation protocol. However, he did a little trick to the flask itself. He made what we call a swan neck in the flask. So he would take the neck of the flask and he would heat it in a flame, and then he took a pair of pliers and he stretched that neck out, and if you notice, it has kind of an S curve in it where it goes up and then down and then back up again. And then he would boil that broth for, I believe it was an hour, and he would observe that after letting it sit out, no contamination occurred of that tube. Now, if he did the same thing, he brought a flask up to boiling in the bottom there, and then he let it sit out and no contamination occurred, it was fine. And then when he broke that neck off, spoilage would occur. And this is from the text in the textbook, and it says, the curve of the flask prevents outside air from entering the flask and no contamination occurs. This is actually false, and this is really important for you to know as a student. This is not what's taking place here. As we recall in the previous experiments, the issue with the spontaneous generation people was that you weren't allowing outside air to have access. Actually, what he was able to do here was not prevent microbes from getting inside of the tube, or not prevent air from getting inside. The key was this allowed air to get through, but in the process of going through these curves, the microbes would get deposited here because barring any strong turbulent air forces, microbes, just like everything else on planet Earth, are governed by gravity. So they will fall to the bottom and also stick to the sides of the glass. And we still have his swan neck flasks in museums today that are still sterile, which is a testament to this experiment, a living testament demonstrating that spontaneous generation was a false hypothesis. So now we've covered briefly the development of observing microbes, the development of demonstrating biogenesis and disproving spontaneous generation, and a little bit about the scientific methods and the rigorous science that had to go behind that. Now, in parallel, let's talk a little bit about the germ theory of disease. Let's begin this conversation with Ignaz Semmelweis in 1847. So he demonstrated the support for what we call the germ theory of disease. That is, that some diseases are caused by germs with hand washing practices. And what he observed was that women who were treated by doctors as opposed to midwives while giving birth were much less likely to contract infections and die. You see, he observed that doctors who performed autopsies on the dead and then immediately go to assist with birth had a higher rate of infections and death with their patients. Therefore, he proposed that washing hands with chlorinated lime water in between patients would reduce the transmission rate of disease. In 1867, Joseph Lister, you might know him for Listerine. Oh, and by the way, Louis Pasteur invented pasteurization. Joseph Lister implemented early aseptic techniques. Aseptic techniques were to avoid sepsis in patients. And he further supported the germ theory of disease. So he picked up on Ignaz Semmelweis's tips about hand washing, and he insisted that surgeons use extreme cleanliness and wash with phenol spray during surgery. At this time, 50% of surgical patients were dying from post-op infections. Now, this was extremely controversial, and many doctors fought tooth and nail against Joseph Lister because what he was basically saying was that doctors were killing their own patients. But thankfully, eventually, science and reason won out on this one. Okay, moving along. In 1876, Robert Koch discovered that Bacillus anthracis, a bacterium, was the cause of anthrax. He did so by isolating it from the blood of multiple infected cattle. And in 1928, Alexander Fleming had a wonderful discovery. He went on vacation and came back to his lab, and his lab was a little messy, and he had some old plates of Staphylococci sitting out. This is a bacterium. And while he was cleaning up, he observed that there was a mold contamination growing on one of his plates. And that's kind of like you observe here in this bottom left-hand photo. And what was fascinating about this mold, which was a Penicillium notatum, was that around the mold, if you look at this mold here, There was no growth of bacterium, so this streak is the bacterium growing, but the bacterium was unable to grow around the mold. And so he discovered that Penicillium mold has antimicrobial properties. And that's where we derived some of our first antibiotics in modern science, penicillin. Today we've come a long way since then. Now we have developed aseptic techniques. We have food handling and sterilization. We have our vaccines. We have antibiotics, antivirals, and antifungals. Also, along with that, we have some resistance. And we have microbial biologics. Microbial biologics meaning drugs that are created from microbes themselves. And no discussion of the history of microbiology would be complete without also discussing the modern phylogenetics movement. So, taxonomy in biology is the science of naming, defining, and classifying groups of biological organisms on the basis of shared characteristics. I think we're all pretty familiar with that. Some of us have heard of the Five Kingdoms, which is now an outdated, outmoded form of referring to different biological organisms by breaking them down into groups. Because in the 1970s, a scientist named Carl Woese developed a method of studying genetics to determine the evolutionary relationship between organisms. He sequenced the genes of a piece of nucleic acid, which is a relative to the DNA, called ribosomal RNA. We'll get into what ribosomal RNA is in the future, but he chose this RNA for two main reasons. The first is they are an ancient protein that is present in all living organisms. So ribosomal RNA codes for the part of the ribosomes, and these ribosomes are required by all life forms. So they're present in all organisms, and they trace back very, very far into the lineage. The second thing is that they're pretty well optimized, and so therefore they're what scientists call highly conserved DNA sequences. That is that their sequences change very slowly over time, because for microbes, if it's not broke, they're not going to fix it. And changing it can have dramatic effects on the way that organism operates, often in detrimental ways. In this way, we can look at that sequence of RNA, of what we call base pairs, or the code of that RNA, and the little differences will tell us how closely related each organism is to one another. In a way, it's as if you took a book, say like the Holy Bible, and you were able to look at the changes from copying it and determine in order which one is older, which one is newer, and which copies were made from other copies, and which one shared a copy long ago. And that is because if you have an error made from writing this code, this book of life, from one individual to its offspring, then that offspring's offspring should also have that error in its code written down as well. And keep in mind, we are all mutants. So every time one copy is made into a new copy for the next generation, there's about 3 mutations per generation. So all of us have about 3 different mutations or misreadings of our previous generation's book. And what Carl Woese developed was what he called the 3 main domains of life. And those 3 main domains are bacteria, archaea, and eukarya. And this principle works the same way that paternity tests can work, or 23andMe by running your sequence. So you can think of this phylogenetic tree of life as one giant paternity test. Now, the reason why this is so important to microbiology is because of what was discovered and what came about in all of this. For a while there, we focused on what was biggest and what was observable or easily observable, or what was most closely related to us, because we view the world in a very human-centric manner. And so we had this animals group taking up its whole kingdom once upon a time of the five kingdoms, Animalia. But if you zoom in here and look at animals, it's a very small part of the tree of life. Actually, animals are very closely related to fungi, very, very closely related to plants and molds and all of these other little beasties here. And if we look to the other side here, bacteria and archaea, here we see the microscopic world. And it's not just here, of course, it's also on the eukarya tree, but all of these other organisms here are microbes. And this really threw the science community off its kilter here. So we've talked about some of these before. We've talked about the methanogens. So they're in the archaea. We've talked about the bacteria. Here we have cyanobacteria. We have gram positives, and that's how they stain. We'll talk about proteobacteria later, and some of the other ones on this list. Now, as you can see, recent developments in our evolution were animals, fungi, plants, and much further back here, we all had a last universal common ancestor, also referred to as LUCA. And scientists are very interested in these interfaces here. When did archaea and eukarya split? When did the archaea-eukarya lineage split from bacteria?
Checkpoint 1. Who definitely and finally disproved spontaneous generation, and how did he do it?
In Chapter 2, How We See the Invisible World, we'll focus on how scientists viewed microscopic organisms. We will mostly focus on light microscopy because that's enough to handle in itself, and it's the most common type of microscopy. In particular, we'll be looking at bright field, dark field, phase contrast, and fluorescence microscopy. Although there are many other types of light microscopy, we'll briefly just talk about scanning acoustic microscopy, which uses sound waves instead of light waves. Electron microscopy is common and uses beams of electrons to look at very small, whether that's subcellular parts or things like viruses and very small objects. And then we have scanned probe microscopy, which inserts a tiny probe into a specimen and then transmits an electrical current through the probe to map the structure of the specimen. Okay, so we're going to talk about the anatomy of the compound light microscope. It's called the compound light microscope because it uses visible light and because there are two lenses in it, two main lens types in the compound light microscope. The first and most obvious one is the ocular lens, and this is the eyepiece, which remagnifies the image formed by the objective lens. The objective lenses are down here. They are the initial primary lenses that magnify the specimen. And let's hop over here and look at figure B, which will give an overview of how the light travels through the microscope, through the specimen, and to your eyes. So down here at the bottom, we have the illuminator. This is the origin of the light source. Light travels up through the microscope through a condenser lens, which focuses the rays of light into a narrow beam that crosses the specimen. So then these light waves pass and cross each other and go to the objective lens right here in the middle. So your specimen is laid on this stage. The light then travels up, gets refracted back and forth into this prism, and eventually is straightened out and comes through these ocular lenses right here. Now, in the process of all of this, the image actually gets turned, gets inverted. And so you're actually looking at kind of the mirror image of that specimen itself, which can be a little confusing because that means when you move this stage left and right, you actually go in the opposite direction than what you're seeing through the ocular lenses. So now that we've done that, let's kind of list the different parts of the anatomy of the microscope. So again, let's start here on figure B, which shows the path of the light. At the beginning, we have the base with the source of the illumination here, which is called the illuminator. The condenser lenses here, which focus the light onto the specimen. The specimen goes here. The specimen isn't part of the microscope, that's just where it goes. The objective lenses are above that magnify the image. We have the body tube here, and then the prism and the ocular lenses. Now let's look at it in a realistic picture, and we'll show the different mechanism and parts that are included in this part A, in this figure A. So if we start from the bottom and work our way up, the very bottom, you will find that there's some sort of a adjuster of the light intensity down here. And above that, you will have what are called focusing knobs. So there'll be one on this side and one on the other side, and they're both the same on either side. They have the same function. The focusing knobs move the stage up here, up and down. And the stage is what holds the specimen itself. We have two types of knobs. We have a coarse focusing knob, and that's this larger knob. And we have a fine focusing knob, which is this inner smaller knob. And can anyone guess what those two knobs do? What's the difference between a fine focusing knob and a coarse focusing knob? The answer is the coarse focusing knob moves the stage quicker, and as a result, it allows you to kind of move in and out of focus more quickly. The fine focusing knob is for fine tuning it. So we're dealing with a very small amounts of changes result in very big differences in the focus that you actually end up seeing. So the fine focusing knob allows you to fine tune whatever you can't do with the coarse focusing knob. Okay, so let's keep moving up here. Yes, we also have the illuminator source here. Here is the diaphragm. The diaphragm, it controls the amount of light entering the condenser. The diaphragm is like how your eyes will dilate in order to control the amount of light that goes through your eyes. So when you first wake up, things might be very bright because you've adjusted your eyes to let in just a narrow amount of light. So this diaphragm can control the amount of light that enters the condenser. You typically want more light for a specimen that's thicker, or if there's more mass on that specimen, so that you get a less dark image. Here's the condenser right up above it, and the condenser, once again, it focuses the light onto the specimen. It doesn't focus the specimen so that it's in focus on your eyes. It just tunes the light to the specimen itself. Next is the stage. Now the stage is where the specimen performs. It's where you put your slide here. And typically, it's hard to see in this image, but there's usually some sort of a specimen holder or specimen arm that flips out and kind of holds that glass there snugly in place on the stage. Next are the objective lenses. Here we can see the different lenses which have different magnifications. So you can increase the magnification level. These objective lenses are held to what we call the nose piece. The nose piece is this black rim you see here, and it holds all of the different objective lenses. So this rotates around in a circle so you can rotate between different magnifications. Typically, you start from a very low magnification and then find your specimen through the microscope. Once it's found, you fine-tune that specimen, and then you will rotate this lens to a higher magnification. So you might go from 10x to 40x to 60x to 100x. So you keep increasing the magnification, and each time you readjust the focus so that it remains in focus all the way up to the highest power. Okay, so next we have the arm. That's just this physical part right here that kind of holds the top piece of the microscope to the bottom half of the microscope. The body of tube we've already discussed, and the ocular lenses are at the top. The ocular lenses are almost always a 10x ocular lens. So we have a variety on the nose piece here, but the ocular typically is always a 10x. Okay, here is a zoomed in image of kind of the business end of the microscope. If we look, we can see we have the glass slide here. So not depicted here would be the stage that holds the glass slide. So if we work from the bottom up, we have our light source coming in. The left and the rights are just designated red and blue, so you can keep track of them. Here we have the iris, and the iris is a movable piece, and it can shrink. These two walls come in and shrink around it, or they can expand in order to increase the funnel of light that comes through here. Here's the condenser, which can be moved up and down in order to control the angle at which the light comes in here and focuses on your specimen. So you want a razor sharp focus of light that comes here onto your specimen. So again, here's a glass slide. The specimen is always on the top of the glass slide. So if it's upside down and the specimen is on the bottom side of the glass slide, then you're actually viewing this incorrectly. You're going to rub off that material off of there and your microscope won't work properly. So always make sure that whatever you fixed to that glass slide, it is on the top of that slide. Then we have some space that will occur naturally between the specimen and the lens here, the primary lens. And then, yes, that light will pass through and be bounced around until it reaches your eyes of the ocular lens. What this is so important to demonstrate is how refraction works in a microscope. Refraction is pretty simple. All refraction is bending of light. This happens naturally in, for example, a rainbow that you see, those water droplets bend the light, and the different wavelengths of light get bent at different angles. And that's why you see visible light being broken down into the different colors. Now here, glass has a similar effect on light. And so we have to take that into consideration when we're trying to view an object. The degree at which light bends as it passes through a particular material, whether that's air, whether that's water, whether that's glass or oil, whatever it is, that degree is referred to as the refractive index. So it's how much light bends as it passes through a particular material. If we look at this graph or this diagram, we can see that light through each one of these materials bends at a different angle and has a different trajectory before it reaches the objective lens. So the condenser bends the light, and that's great. That's what we want it to do. We bend it towards the specimen, and then it reaches and passes the specimen as we want it. But from here, it gets a little messy. If we let it pass through just air, the air is going to refract the light at a different angle than the glass did because it has a different refractive index, and there's a decent amount of distance between that specimen and the objective lens. As a consequence, when you're using very high magnifications, unfortunately, that amount of bending is no bueno. it will put the specimen, you'll never be able to get the specimen into focus, and it will always be a blurry image. Now, scientists have developed a workaround for this. After you pass your 10x objective and your 40x objective, and you're moving to a high power objective lens, you will take a droplet of what they call immersion oil, and you're going to place a very small little dab of oil onto your specimen. Then you're going to move to that next high objective lens, probably a 100x. And if you look at the blue light here, when the immersion oil is present, the immersion oil has a very similar refractive index to the glass. And therefore, the light does not get bent too far, and it reaches the objective lens perfectly well and reaches your eye. So to summarize, oil must be used to ensure that the light is directed from the source to the objective lens. So what do we mean when we say that a lens is 10x magnification? What that means is that it's 10 times the magnification at which you would see with the naked eye. Same with 40x, 100x. 100x is 100 times what you would see with the naked eye. So when we're looking through a compound microscope, we need to keep in mind that there are actually two lenses that are both magnifying the view of the specimen. We have the objective lens, which is this first set of lenses here, and we have the ocular lens, which is this lens we look up through here. Now take a moment and see if you can remember what the magnification typically is for an ocular lens. Hopefully you said that it was a 10x. Yes, typically ocular lenses are set to 10x magnification. So, how do we figure out the total magnification? It's a very simple calculation where we take the objective lens power and we multiply it by the ocular lens power. So if you have an objective lens that's set to 10x and an ocular lens set to 10x, what's the total magnification? That's right, it's 100x magnification.
Checkpoint 2. If the ocular lens power is 10x and the microscope is set to the high power objective lens 40x, what is the total magnification of the specimen?
Finally, let's discuss resolution. Resolution is very important in making really good microscopic images. What resolution is the ability to distinguish between 2 points. So depending on what device you're using right now, you may or may not be able to distinguish the 2 periods I have listed below that. If you can, you have higher resolution than someone who cannot distinguish 2 periods in that line. Ultimately, what this means is that the lower resolution you have, the fuzzier the image becomes. And the higher the resolution, the sharper the image is. And there are 2 factors that affect this. The first being wavelength of the light. Light rays can come in different wavelengths, and we'll show that here in the next slide. And the shorter the wavelength is, the more likely, the more capable you will be to determine if there's that 1 period there or 2 periods there. Number 2 is the numerical aperture. So we've already discussed magnification, which is the ability to increase in size, the real size of an object against the naked eye. Numerical aperture is the lens's physical ability to gather light. And we're not going to get into the mathematics of that, but the more light that it can gather, the better. So with numerical aperture, the higher the numerical aperture, the better the resolution. So what do we mean when we say the wavelength of light? Each type of light has certain features about it. So here's a cartoon depiction of a light wave, and we can see here's the wave. And the waves have a crest, or the highest point, and a trough, or the lowest point in the wave. And the distance between the trough and the crest is the amplitude, and that's how intense that beam of that ray of light is. It's kind of like with sound, the higher the intensity of sound, the louder it is. That doesn't really change the innate characteristic of that light. However, what does is the wavelength. The wavelength is what's unique to each type of light, and that is the distance between the crest and the crest, or equally so, the distance between the trough and the next trough. And here is a depiction of what that looks like. So we have a whole variety of different sorts of electromagnetic wavelengths to concern ourselves with. Us as humans, we are typically most concerned with the visible light electromagnetic spectrum. But if we look here, maybe if we start as a good reference is FM, which is a 1 meter wavelength. And then here we have radar, which is smaller than that. This is 10 to the negative 3. So that's 1/1000th of a meter in size. So that's a decicentmilla, a millimeter in length. Still visible with the naked eye. You can see a millimeter on a ruler. Here we get into infrared. Infrared is somewhere between 1/10,000th to 100,000th of a meter. And then we get into the nanometer space for us. So we see about 0.78 micrometers, or 780 nanometer light, all the way down to 380 nanometers of light. So it's too small for us to see the wavelengths, but we can see what sort of light that, what color that creates in our own mind's eye. We have many other types. We have ultraviolet rays as well. We have X-rays and gamma rays. But since we're using a microscope to view things with our own eyes, we have to use the visible light spectrum. So I want you to go ahead and make an educated guess as to what type of light in the visible light spectrum we would want to use to have a higher resolution. Now, if you said a shorter wavelength, you would be correct. A shorter wavelength is going to have less overlap between two very small points of an object than a higher wavelength. So one thing we can do to increase our resolution is to use something in the visible light spectrum that has a shorter wavelength. Now, let's not run away from these numbers just yet. Remember how we discussed that bacterium could be roughly 1 micrometer in size? Well, if we look here at the very end of what we're capable of viewing with the naked eye, or with a compound microscope with a visible light, that's around 0.38 micrometers. So if you see an image that is at high power magnification, let's say the ocular lens is set all the way to 100x, or sorry, 10x is the ocular lens, but the objective lens is set all the way to 100x. So you're getting about a 1000x magnification. And you have a really good aperture of your lens, and you have a really short wavelength. The smallest things you're going to see is about 0.38 micrometers in size, or about 1/3 the size of a bacterium. So we're not going to be able to see things much smaller than bacterium, and we're lucky if we can see different parts of a bacterium, such as maybe its nucleus inside, or its flagella, or its cell wall. These things are very difficult to observe through a light microscope. So if you're presented with a question and you have to determine on full magnification of a microscope and you can just barely see these little organisms in the microscope. You're probably not dealing with protists or protozoa or anything like that. Rather, you're probably dealing with bacterium. Also, on the other hand, if you're just barely able to see something at the highest magnification possible in the microscope, it's not going to be a virus typically, because viruses are much smaller. They're on the very far end of the range, and we need something like a electron microscope to observe viruses. Okay, so let's talk about the different types of light microscopes that are out there. The first one, and the simplest one, is the bright field microscope. This uses visible light to illuminate a specimen. And because if you imagine the light passing up through the condenser, the diaphragm, onto your specimen, the specimen's going to block out some of the light, and anywhere where there isn't a specimen present or no objects present on that glass slide will appear bright. So the specimen is actually dark, surrounded by a bright field or a bright background. The resolution limit here, again, if you're amazingly good with your resolution, is 0.2 micrometers. In practice, that's not actually a very good resolution for the reasons I outlined with wavelengths and what we can see in the visible light spectrum. Okay, so dark field microscopy uses a special condenser to block light from entering the objective lens. And it has a small disc, as we see here, that's about 1 centimeter wide and blocks most of the light. So let's take a moment, and I want you to think to yourself, why is this called a dark field? If you're looking at this image, what are you likely to see as bright and what are you likely to see as dark? You should have said that you believe that the field or the background is going to be dark and the specimen is what's going to appear illuminated. And that has to do with that little disc that's there. So what happens is this light comes around and the light the light that is reflected or bounced off of the specimen actually is what enters the objective lens, and that's what you end up seeing. And this is really useful for visualizing organisms that can't be seen using bright field microscopy. Typically, those are organisms that don't stain very well. So this could be live organisms or just organisms that you don't end up using a stain for one reason or another. And here is an example of a bright field microscopy. And we can see this is a paramecium. And if we go ahead and take a look at the scale bar here, we can see it's 25 micrometers in length from here to here. So this is probably about 8 to 10 times the size that. So this would confirm that it's about the correct size for a paramecium, around 250-ish, 200 micrometers. If you'll recall, we're looking at bacterium on somewhere in the scale of 1 to 5, 1 to 8 micrometers. So another clue is we can see all of this, what we call ultrastructure, or the inner parts of this cell. So there's some sort of other organelles and things that we are seeing inside of the cell that you would not be able to see. You would see just little specks if it was bacteria, like the other specks around here. So again, here we have a specimen that appears bright against a dark background, and this allows for visualization of organisms that can't be stained. Okay, phase contrast is the next one. There are two types of light that enter the objective lens. There's a direct light from the light source, and then there's also the reflected or diffracted light from the specimen itself. And there's not any need to fix or stain with this sort of phase contrast microscopy. And it provides these very rich, deeper visualizations of a specimen. And it allows for visualization of internal cellular structures. So you can see that all of these internal structures are much brighter or darker compared to dark field and bright field. We're not going to get into too much detail about how these phase contrast microscopes work. But you will need to know where it gets its name from. Where does the name phase contrast come from? And that again has to do with these wavelengths of light. So remember how we said that if the light is reflected or diffracted or refracted, the light bends. And when it bends, it has to travel a farther distance in order to reach your eye, because in the phase contrast, it gets bent back. And what happens is, because it travels a longer distance, the wavelengths that we see here get shifted. So if we look here, these are both in phase, these two wavelengths, right? But say in the second part of the image, say that one of these wavelengths, it takes a reroute, and so it gets bent and slows down. And then this causes the wavelength to be out of phase of other wavelengths that didn't get bent. In a phase contrast microscope, these get added back together. And if you were to add these two wavelengths together, the trough of this first wavelength and the crest of the second wavelength will therefore cancel each other out. So a negative plus a positive of the same intensity will equal zero. The same is true when we have a crest on this part of the wavelength and a trough on this part of the wavelength. The combination of those two is equal to zero. And so therefore, this is called destructive interference. And so you'll end up not seeing any light at all where these two converge. On the other hand, in a phase contrast where you have two wavelengths that are still in sync with each other, naturally they will have constructive interference, and they will increase in their intensity, so you'll see them even brighter. And that's what causes some of the light to be very bright and the light that gets refracted away to be very dark. That's what causes the deeper visualization. So for the exam and for the future, you just need to know that phase contrast means that you have light that gets bent or refracted away, or reflected or diffracted away, and that causes the light to become out of phase. And those out of phase light beams have destructive interference, which decreases the intensity of light. And those that do not are in phase with one another, and so you see a constructive interference where they become even brighter. Another fun type of microscopy is fluorescence microscopy. And this again uses the visible light, sometimes even all the way to those really short wavelengths, which are called ultraviolet. So that would be a little bit smaller than what we're able to pick up with our eyes. And it makes a very, very brilliant images. And the way it does this is it uses naturally fluorescent organisms, or organisms that are stained with what we call fluorochromes. These are fluorescent dyes. So a light source has a specific wavelength of light, and this can be ultraviolet, for example, that stimulates these fluorescent markers to fluoresce. Next is a filter between the specimen and the ocular lens that blocks all of this light that was emitted from your light source, but it allows the color from the fluorescent marker to be observed. So the light that you're seeing coming through this image is not the light from the light source itself, but it's a light that's being emitted from the light source, just like glow sticks. If you hold them up to a light or a UV light, they get charged, and then you can turn off the light and they will glow because they've absorbed the light, and then they fluoresce a new light of a different wavelength. This is especially useful when you're trying to see certain things like chlorophyll pigments, carotenoids. There's a lot of pigments out there, especially in photosynthetic organisms, that can absorb one wavelength and then give off another color at a different wavelength. So you can really observe if those structures are present. The other thing you can do is you can introduce these proteins or these compounds by genetically engineering your organism so that you can see if it's present or not in the organism. And sometimes only under certain conditions will that gene be expressed. So it's a very powerful tool. And what ends up happening is you have a sample that appears a bright color against a darker background. Okay, so here once again, we're just going to show you fluorescence. As I said, here's the ultraviolet spectrum here. So you can use a spectrum that's not visible with the naked eye, but it has a lot of energy and it is absorbed by those fluorescent fluorochromes or proteins or other things that fluoresce inside of the cell and give off a light that's visible in the visible spectrum. One example of this is what we call green fluorescent protein, or GFP. It's a very common fluorescent marker. It's a small gene. I think it's about 300 base pairs in length is all, which in terms of genes is about half the size of the average gene. And it's often used as a tag for bacterium. So in the image you see off to the right, GFP tagged a cyanobacterium, and you can see that they glow a brilliant green color. This is the GFP protein present inside of the cells. So this image is first exposed with a UV light onto the specimen, and then the specimens themselves, they give off a brilliant green fluorescent color, and then a filter is placed between the specimen and the ocular lens. And that filter specifically blocks out UV light, but allows the green to pass. So as a consequence, we're only seeing the green color. Now there's so much GFP in this image that we get a little bit of background green, but that's okay. And this image right here, Candidatus promethoarchaeum centrophicum, if you will recall, this is that organism that we showed in the previous lecture that was closely related to bacteria and eukarya, that were really close to that diversion point between these organisms. And it actually is called centrophicum because there's a lot of symbiotic relationships between it and the presence of other organisms. And each one of those organisms are very hard to see because they're little bacterium. But by using fluorescence microscopy, we can pick up fluorescent signals from different bacterium, and we can use a computer software to generate the populations of bacterium that exist in that culture. So this brings us to our discussion of how samples of microbes can be prepared for light microscopy. There are two things your mother never wanted you to do, and that was staining and smearing. But in microbiology, we do a lot of staining and smearing. So what is staining and smearing? Well, staining is a way of coloring a specimen with a specific dye that emphasizes certain structures. However, before staining, a specimen must be attached, which we call fixing, to a slide in a thin layer called a smear. This is the typical process at which a smear is done. So if we have a liquid media, these are like broth cultures that you often see growing in tubes, you'll use this tool right here. This is an inoculation loop. See the loop on the end? And it's kind of like one of those little plastic bubble blowing things that you might use. It has a handle, it has a stem, and a little eyelet there at the end. They're typically metal, however, and that's so that you can sterilize them with heat. So what you do is you would sterilize this inoculation loop, you would dip it into the broth culture, and you would take out a sample of liquid film which would form around the loop. And then you would take a glass slide and you would draw with your magic marker, probably on the back side is best so that you don't damage the marker you made. And you would start dabbing 1 to 2 dabs, 2 loopfuls of liquid to cover this entire area. And you should just barely see some wispiness on here. Imagine, again, these microbes are only 1 micrometer in size. So if you see more than wispiness, you have layers of layers of layers of bacterium, and you won't be able to see what's going on underneath the microscope. It'll just look like blobs. So after you have that done, you're going to let this smear just air dry. Okay, so you're going to just set it down on your desk and let that dry until it looks nice and dry and there's not any water remaining. Students often get a little impatient with this step, but you just need to be patient with it, because in the next step, if you go ahead and do the next step before it's dry, you'll cause boiling to occur and you'll cause the cells to lyse and break open, and then you won't be able to see any cells when you go to view it under the microscope. A similar process can take place from solid media. So sometimes we grow our cultures on a solid medium, typically what's called agar plates, which we'll discuss later. And from this solid media, you again would take just a teeny little scoop, 1 little loopful of it, and you place it there. Now a loopful has thousands and thousands and thousands of bacteria, so don't feel like you need to get a particularly large loopful. Just a little bit will do the trick. And then you're going to spread it around. Here they've spread it around with an inoculating needle. That's great. You can also use that loop to spread it around, just as long as it's nice and thin. You should have to hold this up to the light to visualize that smear. That's how little you should have on there. Again, once you smear it out into a nice even smear, go ahead and let it air dry, and this can take several minutes to accomplish. After you've allowed it to completely dry, you're going to pass this across a Bunsen burner. Now it looks like this person is just holding it over the flame. That's not what you want to do. You just want to heat it up a little bit. And what that heating does is fix those cells to the slide. So you're not trying to roast them. This isn't a barbecue, that you're not making s'mores or anything like that. You're just going to quickly pass it a couple of times to warm up that glass slide. Sometimes we use a Bacticinerator, which is essentially just a little ceramic heating element, and that's not quite as aggressive as an open flame, and so you'll hold it against that heating element for maybe 20 seconds, 30 seconds, just enough to kind of warm that glass slide up just a bit. You want to use a clothespin to do this, or a pair of pliers or something, so that you don't burn your fingers. And you also want to make sure that if you look under the microscope and you don't see any bacteria, that might have been for 2 reasons. You did this for too long and you destroyed your cells, or you did it for too little and during the staining process you washed away the cells because they weren't fixed. So it takes a little bit of troubleshooting to get the hang of, and it's a little bit tricky. Okay, now let's get to the staining part. Stains are composed of different parts. Stains are salts that are composed of a positive and negative ion. So here you might need to do a little bit of a review of what a positive and a negative ion are. A negative ion has electrons, an excess of electrons versus the number of protons. And that just makes the atoms in that molecule more negatively charged. A positive ion has fewer electrons than it should have, and so that makes it positively charged. It's lacking in those electrons which have a negative charge to them. So, to summarize, either the positive or negative ion is a chromophore. The chromophore is the part of a stain that is colored. Remember that. The chromophore is the part of the stain that is colored. In basic dyes, the chromophore is the positive ion. In basic dyes, the chromophore is the positive ion. In acidic dyes, the chromophore is the negative ion. Bacteria are typically negatively charged on the outside of their cell walls. And you should know by now that in electricity and in magnetism, opposites attract. So if the bacteria is typically negatively charged, typically, do you want to use a basic dye or an acidic dye to bind with it? You should have thought that a basic dye would be more appropriate, and that is because the positive ion chromophore will be attracted to the negatively charged cell wall. However, sometimes you want to stain the background. This gets a little confusing because a background stain is called a negative stain, and staining the bacterium is called a positive stain. You can remember this because a positive stain has a positive ion normally, and it is a basic dye. A negative stain has a negative ion and for a chromophore, and it's an acidic dye. The reason why acidic dyes don't stain the bacterium is because they're both negatively charged. Therefore, you will have some residual that sticks to the glass, but you should have clear zones where the bacteria is. All right, so there are three different kinds of staining techniques that we can do. The first and most simplest one is simple staining. It uses just a single basic dye. And remember, basic dyes are going to attach to the outside of the cells. This highlights the entire microorganism and allows us to visualize the cell shape and sometimes internal structures. In the example, you can see methylene blue provide a simple stain of human cheek cells. The next one is a differential stain. Differential stains tend to be much more complex. They involve multiple dyes, and they are used to distinguish between two different types of bacteria or cells. The most common of which are gram stains and acid-fast stains. In the example image, you can see this is a gram stain, again that should be capitalized, gram, of Lactobacillus crispatus. And it's purple, so it is what we call a gram-positive stain. Here are the general steps in the gram stain. So step number one is the addition of the primary stain. In the gram stain, it is crystal violet. Crystal violet dyes all of the cells a purple color. Now, it dyes all of the cells regardless of them being gram-negative or gram-positive in this first step. So any cell present, you can see it's purple. Step number two is the addition of a mordant. A mordant is an ion that binds to that dye and it helps it to hold itself in place better. Sometimes we might even use heat as a mordant. In this example, it's a chemical mordant, iodine. This creates what we call the crystal violet iodine complex. The next step in this process is to get rid of the primary stain so that we can apply our contrasting stain. So here we have an alcohol wash. We apply alcohol for just a few seconds in order to cause the decolorizing to occur. Those that are gram-negative will lose their dye readily. However, those that are gram-positive will retain the dye. So at this point, we should have our gram-positives purple and our gram-negatives a colorless image. Lastly is the secondary stain or counter stain. In gram stains, it's safranin, and its color is a pink color, and any of those bacterium that no longer have a color will be pink. All right, so if we look at our actual example here, We'll see we have two different bacterium. That's obvious because one of them are bacillus shaped and one of them are coccus shaped. And in this image, we have, which one is the gram-positive? You should have said the coccus. The coccus is, it looks kind of blue in this image, but it's kind of a bluish, purplish color. Then you have the gram-negatives, and those are the rods. So it's a nice little image. And why does this occur? This occurs because the bacteria with a very thick layer of peptidoglycan in their cell walls retains the crystal violet during the decolorizing step. However, the bacteria with a thin layer of peptidoglycan in their cell wall readily give up that color during the decolorization step. So, those gram-positives retain the primary stain and are purple in color, and they're called, therefore, gram-positive. The gram-negatives lose the primary stain during washing, take up the counter stain, and therefore are pink in color, and are therefore called gram-negative. So why is this important? Well, obviously it allows an examiner to differentiate between two major groups of organisms, or to start to classify what organism they might have. So it allows an examiner to diagnose a disease, a contaminant, or an environmental sample. Also, the thickness of the cell wall has consequences for what type of antibiotic is effective against the bacterial species. Certain antibiotics work for gram-positive species, while others work for gram-negative species. Okay, the next stain we're going to discuss are acid-fast stains. Acid-fast stains bind only to bacteria that have waxy material in their cell walls. This includes bacteria from the genus Mycobacterium, which are bacterium such as M. tuberculosis, which causes tuberculosis, and M. leprae, which causes leprosy. And also Nocardia, which some of which are non-pathogenic and others are weakly pathogenic to those who are immunocompromised. So this is how the acid-fast stain is conducted. Once again, the primary stain is added first. This is carbol fusion, and it is applied to a fixed smear and colors all cells pink. Then we're using heat as a mordant this time. So the slide is gently heated. In this image right here, we have a little heater and a water bath that the slide is placed over. So that's just so it doesn't get too dried out in this process of chemically using a heat mordant. Then we have a decolorizer. Of course, we want to be able to differentiate, so we need to get rid of that primary stain where it doesn't belong. So we use an acid alcohol decolorizer. And then finally, we add our counter stain. In an acid-fast stain, the counter stain is methylene blue. And this stain stains all cells without a waxy cell wall. So a couple of things to note here. This is kind of the opposite of a gram stain. Remember that a gram stain, the gram positives were purple and the gram negatives were pink. Well, in this example, carbol fusion is pink and methylene blue is purple. So the counter stain is actually the purple one and the primary is the pink. Another thing to note is, why do we use that heat? Well, we use that heat because these waxy cell walls are very difficult to penetrate. And there's a good reason for that. It's a defense mechanism for the Mycobacterium. And so once again, those cells that do not have that waxy cell wall, they readily give up that decolorizer. So it's kind of the same as a gram-positive in this mechanism of being able to retain the primary stain better than the negatives.
Checkpoint 3. Interpret the gram stain below. Is a circled bacteria gram-positive or gram-negative? Checkpoint 4. Interpret the acid-fast stain below. Which letter, A or B, could be M. tuberculosis?
Okay, so some microbes contain a protective coating called a capsule, which indicates virulence, which is the degree to which a pathogen is disease causing. These capsules do not take up most dyes and appear as an unstained halo around the cell when stained with India ink or necrosin. So, this is a type of special staining that is done, and it's referred to as a negative staining because the background is stained and the cell wall itself is not stained. Okay, next one are endospore stainings. So endospores they're kind of like bomb shelters for bacterium. They are very resistant structures. They are dormant cells that can arise in certain bacteria under stressful environmental conditions. So the staining requires the Schaefer-Fulton method. The primary stain is malachite green, and it uses heat and a decolorizer as water and a counterstain of safranin. The consequence of which, as you can see in this image, is that the endospore is stained a green color from the malachite green, and then the counterstain is the pink. So those endospores hold on to that primary stain really well and appear to be a greenish color. Okay, next up are flagella staining. So flagella are these little appendages that come off of microbes that typically help with things like motility. And an unstained flagella is extremely small and it's hard to be seen with a light microscope. We can use a carbonyl fusion and a mordant that coats the flagella until a thick enough coat is applied to be visualized.
Checkpoint 5. Place the steps of a differential stain in the correct order: mordant, decolorizer, counter stain, and primary stain. Checkpoint 6. Would endospore staining be classified as a simple stain or a differential stain, and why? And that is the end of chapter 2. We'll see you next time when we discuss the different parts of the cell.